Our next speaker is Dr. Eliza Tsou, who's an assistant professor in the Division of Rheumatology at the University of Michigan. Her research focuses on understanding the pathogenic mechanisms involved in autoimmune diseases, with specific emphasis on systemic sclerosis and its vascular complications. Throughout her career, Dr. Tsou applies advanced genomics and bioinformatics techniques to identify key pathways and therapeutic targets, aiming at better understanding disease progression and patient responses to treatments. Her lab is particularly interested in investigating mechanisms that contribute to fibrosis and vascular dysfunction in scleroderma using both experimental models and patient-driven derived samples. And the goal of her research is to identify novel therapeutic strategies for scleroderma. So please join me in welcoming Dr. Tsou.
Thank you, Ahmed. It is my great pleasure to be here to share our research with you. I had the privilege of interacting with Dr. Goldstein a few years ago when we realized that we're dealing with a vascular aging phenotype in our disease. In our meetings, Dr. Goldstein struck me as a genuinely nice person, really passionate about his research, and he introduced me to the M-BoCA community. I really appreciate the time that he spent with me as a junior faculty, and he will truly be missed. Today, I thought I will walk you down the journey of how we stumbled into the world of vascular senescence in our disease, and I welcome any helpful suggestions and comments from this group of experts in cardiovascular aging. So I come from the Division of Rheumatology, and we study autoimmune diseases. The study of interest of mine is called systemic sclerosis, or scleroderma, and this is a multi-system disorder characterized by immune cell activation, vascular dysfunction, as well as tissue fibrosis. In Greek, sclero means thicken, derma means skin. So on the outside, these patients do have a lot of skin involvement. We call skin fibrosis. However, this fibrosis process can actually be spread into the internal organs, and it's these internal organ involvement that causes a high mortality rate of this disease. This is a highly heterogeneous disease with a lot of different subsets. We typically recruit patients with diffuse cutaneous scleroderma because this is the most severe form of the disease with more internal organ involvement. Treatment for these patients are fairly limited, mainly just treating the symptoms but not the underlying cause. And so in this busy slide, this summarizes some of the key features of what we know about this disease. There are the three major components that I mentioned: autoimmunity, inflammation, vascular dysfunction, as well as tissue fibrosis. Tissue damage usually happens at the blood vessel, and this will cause these endothelial cells to become abnormal, and they will form these leaky blood vessels that will allow these activated immune cells to infiltrate into the tissue. Once in the tissue, these cells can produce a lot of pro-inflammatory mediators that can make this tissue very inflamed. And in this environment, these resident fibroblasts, which can come from different sources, can be activated and further transformed into myofibroblasts. And these cells produce a lot of extracellular matrix that will lead to tissue fibrosis. In our lab, we study the blood vessels of this disease, and this slide summarizes some of the vascular manifestations we see in these patients. One of the most prominent features is the Raynaud's phenomenon, which is shown here on the top right, where there's changes in the fingers, and this is due to disruption of the blood flow. This usually happens when there's drastic changes in temperature, such as the fall weather we're experiencing right now in Michigan. Another feature that they have is called nail capillary changes, and this is shown here. These capillaries and their fingernails show these dilated blood vessels, sometimes avascular areas, and you can see this is a drastic difference comparing to the capillary beds from a healthy control. Other manifestations include telangiectasia, digital ulcers, pulmonary arterial hypertension, and most severe form is scleroderma renal crisis. For morphological changes, we see two different types of vasculopathy in these patients. One is called fibroproliferative, and usually happens in the small arteries. In this case, in this biopsy, you see complete occlusion of the lumen being replaced by these ingrowing, proliferating vascular cells. On the other hand, there's a total opposite of fibroproliferative. There's destructive vasculopathy. This usually happens in the capillaries. You can see these thin layer of the cells, sometimes complete dropout of the endothelial cells, and manifesting into this phenotype that we see with these leaky blood vessels and immune cell infiltration. And so vasculopathy and scleroderma is an early and critical event. And this is especially true for the Raynaud's phenomenon I showed you. Usually these patients, this is the first sign of symptom that these patients suffer from, and the Raynaud's happen way before they have tissue fibrosis. Although this is an early and critical event in this disease, the underlying cause and mechanisms still remain poorly understood. So our goal of our lab is to try to bridge this gap in this research, and that trying to find these underlying causes and mechanisms that can explain this vasculopathy that we see in these patients. So in scleroderma research, we are kind of lucky in a way that there's a lot of skin involvement, and skin biopsies are relatively easier to obtain compared to a lung biopsy, for example. So in the clinic, we typically get these small size of skin biopsies from patients. They're typically similar to a size of an eraser you'd find from a pencil, so very small. And from the biopsy, from different compartments of the skin, we can isolate different cell types. From the epidermal layer, we can isolate keratinocytes. From the dermal layer, from these connective tissues, we can isolate fibroblasts. And from the blood vessels, we can isolate endothelial cells. Since our research focuses on blood vessels, I want to spend a little bit of time talking about the vascular endothelial cells we can isolate from skin. This is taking pictures every 4 hours to show you that these cells isolated from patient biopsies do grow in a petri dish. They have a very short lifespan. We can use them up to passage 6. And these cells, in our lab, we call them princess cells because they need a lot of pampering. If you think about the avascular nature of this disease, these endothelial cells are not very happy to begin with in the skin. Once we want to grow them out in a petri dish, they need extra care. And so this is a niche that our lab built, that we were able to isolate these vascular cells and use them in functional assays to try to help us understand scleroderma vasculopathy. So with this valuable resource in hand, we start to characterize these cells over the years. In the next few slides, I want to summarize some of the abnormal phenotypes that we realized from these cells isolated from scleroderma patient skin. The first feature that we looked at is angiogenesis. When we think about endothelial cells, we think about how they build blood vessels, and that's angiogenesis. There's this assay which is very nice. It's called matrix gel 2 formation assay. And so when you plate these cells on this gel-like substance, they will start forming tubes, just like how they're forming blood vessels in the body. You can appreciate on the left, it's a healthy control. On the right is endothelial cells from patients. You can see that these endothelial cells from patients do try to form tubes, but they're not as well established as what we see in a healthy control. And also the nodes where these tubes are coming out are smaller and not as well structured as what we see in a healthy control. So there is definitely impaired angiogenesis in these isolated cells from patients. Another key feature that we looked at is barrier dysfunction. Remember that vasculopathy, destructive vasculopathy I talked about with the capillaries. And so in this case, we can look at VE cadherin, which is stained in purple. And this is an adherence junction protein that kind of seals off where the cells contact. So we can see this very nice staining of purple in these cells isolated from healthy subjects. And you see those purple staining in our scleroderma patient cells, but you can see that it's not really sealing the edges very well when they contact with each other. So what this translates into is that when we do a permeability assay, where we plate these cells on the transwell and put HRP on top and see how much HRP can see through the membrane, we see that these endothelial cell monolayers have higher permeability, allowing more HRP to get through the membrane, suggesting that there is definitely barrier dysfunction in these cells. Another key feature that I think is very important for scleroderma of this disease is that these endothelial cells undergo something called endothelial to mesenchymal transition, ENDO-MT. So what this means is that these vascular cells are having an identity issue. They don't want to be vascular cells anymore. They want to be more of a fibroblast. Maybe it's cooler. And so over time, they kind of lose their vascular markers and phenotype and gain this more mesenchymal fibroblast phenotypes and markers. So one of the ways that we can show this is through staining. On the left is vascular endothelial cells isolated from a healthy controlled skin. And green is VWF, which is a vascular marker. You can see that these cells are happy vascular cells. Prominent vascular markers shown here. However, for scleroderma patients, you can see that the green disappeared and is replaced by this red staining. And this red staining is staining alpha smooth muscle actin, which is a fibroblast marker. So these cells are still vascular cells, but they're switching more of a mesenchymal phenotype. We also have data from the RNA levels showing the same thing. CDH5 is V cadherin. It's significantly lower in our patient-derived cells. Fibrotic markers increase, and also SNI1, SNI2, which are the transcription factors that's driving this endo-MT phenotype, is also significantly elevated in our patient-derived cells. And we also have data from single-cell RNA-seq that supports this, which I'll touch on in the later part of the talk. In addition to phenotypic changes, we also are interested in the chromatin level at an epigenetic level, whether there's a difference there. And so to look at genome-wise changes of chromatin accessibility, we use ATAC-seq, which allows to look at genome-wide changes, whether the chromatin is more condensed or more open. And so through this analysis, we show that in this volcano plot, you can see the chromatin structure is more condensed in our endothelial cells isolated from patient compared to healthy control. And through this sequencing, if we do deep sequencing, we can actually look at transcription factor footprinting. And through this hint ATAC motif analysis, we were able to identify 24 transcription factors that are differentially bound in our patient-derived cells compared to healthy control. And so with those transcription factors, we went on to do a pathway analysis to kind of get a hint of what these transcription factors are trying to do. And all of the pathways are kind of expected, such as this MAD pathway and TGF-beta signaling pathway, since this is a profibrotic mediator, and this hints to the endo-MT results that I showed you earlier with these endos. To our surprise, there's some pathway that popped out that it's the unexpected. One is this telomerase H-TERT transcriptional regulation pathway, and also the direct P53 effector pathway. And these are pathways that are related to aging and also senescence. So this led us to think that maybe this unbiased sequencing approach led us to think about maybe there's an indication of cellular senescence that's present in our patient-derived cells that we've never thought about before. So cellular senescence is a state of stable cell cycle arrest in which the cells are just stuck in there. They don't proliferate, but they don't die. So over time, there will be accumulation of these senescent cells in the body, and once it reaches a threshold, it will probably do some harmful things in our disease. And so under normal physiological conditions, though, having some senescent cells is actually good for you. It's very important for wound healing, for example. So shown here is just some characteristics of typical senescent cells would have, and we're using senescent endothelial cells as an example. One of the key features of a senescent endo can do is that they can induce paracrine senescence through spitting out these proteins we call SASP. This stands for senescence-associated secretory phenotypes. So these are cytokines, chemokines, growth factors that are pro-inflammatory. And these senescent endos can spread their senescence to neighboring cells by spitting out these SASP proteins and infecting neighboring cells to make them become senescent. This is how they spread their senescence signal. On another hand, senescent endos can also do something called autocrine senescence, that kind of reinforcing their senescence phenotype so that they stay in that state. Other key features for cellular senescence include these markers shown here: beta-galactosidase, reactive oxygen species, DNA damage, cell cycle arrest. Some endothelial-specific senescent phenotype, including decrease in eNOS and also decrease in endofunction. So when we stumbled into this field of senescence, we're kind of surprised to find that the field is kind of messy and that there is not a very good way, definite way to define what is a senescent cell. So after a lot of soul searching, we decided to adopt this multi-marker stepwise approach to kind of help us validate that we indeed are looking at cellular senescence in our system, and it's true. So we started from step 1, screening for general senescence, beta-galactosidase, into verification with additional markers. And then we will look for a specific type of senescence, and we think that oxidative stress and DNA damage plays a key role in this disease, since it has been shown before in other cell types. And then we went to verify it with more endothelial-specific senescent phenotype. So this slide summarizes everything that we looked at, and I'll walk you some of the data on the next few slides. So the first experiment that we did is to look at beta-galactosidase. When people think about cellular senescence, they think about beta-galactosidase. And so there are a lot of tools that allows us to do that, and the tool that we chose to use is called Spider Beta-Gal. This is a probe that, when it's free, It does not fluoresce. However, when it enters cells and binds to SA beta gal, they will start to fluoresce. So it's a very good tool for us to use in flow cytometry. And indeed, when we tested in our system, there is significant increase of these SA beta gal in our patient-derived cells compared to healthy control. We also did staining in the Petri dish in the same passage number and an age match control. You can see that in P6, passage 6, these cells do start to show beta-galactosidase staining, and then at passage 7, it's when they crop out. They have a lot of beta-galactosidase staining and with the enlarged cell shape, and that's another indication that these cells are entering senescence sooner than the healthy controls at the same passage. Other markers that we looked at include cell cycle arrest from this list, and you can see that there is indeed increased P21, P53, and P16 levels in our patient-derived cells. And we also have data from our single-cell RNA-seq results to support this. And this got us to think about what are the upstream causes that leads to all of these cell cycle arrest and senescence that we're seeing. And we think that DNA damage plays a role here, and since it has been shown in fibroblasts and PBMCs that it has been indicated in scleroderma, but it has not been done in vascular cells. So this is what we did next. We looked at DNA damage. So when there is a double-strand DNA break, it attracts ATM, and in this case, it will phosphorylate neighboring H2AX, and we call these gamma H2AX. So phospho-ATM and gamma H2AX is what we stain here. You can see in red, these red dots is where the double-strand DNA break is. The green is where phospho-ATM is recruited, and we can overlay them. We can count how many positive dots are there. And by quantification, you can see that there is indeed increased DNA damage in our patient-derived cells. So the next, well, coming back to these SAS proteins that I said, that these are pro-inflammatory proteins that these senescent cells can spit out, and they could do a lot of bad things. And so here we handpicked 4 SAS proteins, reading through the literature and picking them out more pertinent to scleroderma disease pathogenesis. You can see that these handpicked, cherry-picked 4 SAS proteins are all upregulated. This is measured in tissue culture media, so they're spit out from the cells and into the media. And from here, we went on to see whether these SAS proteins that are spit out from the cells can induce paracrine senescence. Remember, these SAS proteins can be taken up by neighboring cells, and perhaps that will lead to senescence in those cells. And so what we did here is we collected culture media from healthy controls and patient-derived cells, and then we used these culture media to culture healthy endos and see whether there is a phenotypic change in these endos that receive the culture media. And for sure, we saw that in these healthy cells that are cultured in scleroderma patient media, you can see that there is increase in expression of P21. We know that this is highly variable, and we're going to repeat this and make sure that it's true. But this suggests that there is indeed that possibility that the secretome of scleroderma patient endos can induce neighboring senescence and other cell types. So all of this so far is in vitro, and we know that a lot of criticism in the senescent field is that you don't know whether you're seeing this due to an in vitro artifact or whether it's true. So in the next few slides, I'm going to show you what we found in skin biopsies. So shown here is staining skin biopsies from either healthy controls or patients with diffuse cutaneous systemic sclerosis. And on the left, you can see the VWF is green. It's the vascular marker, so these are blood vessels. Ki67 is a proliferation marker. And so you can see there's prominent red staining in healthy controls, but not so much in the patient slides. On the other hand, we also stain for P16, which is senescent marker. You can see the senescence marker labels the blood vessel very well. There's colocalization of both. And by quantifying this, we definitely see decrease in proliferation and increase in senescence in the blood vessels in the skin in our patients. We then went on to look at single-cell data. And so this data set is generated in 18 healthy controls and 22 scleroderma patients. These are patients with diffuse cutaneous early disease, so that they're really on their most severe form of disease in the time course of the disease. And in this single-cell data, this is generated in collaboration with dermatology with Dr. Johan Gudjonsson. We identified 12 different cell types from the skin, shown here. And what we did with this data is that we generated a score called the senescence score. And this is by choosing 68 senescent-related genes. This is handpicked by me. This is a homemade list of SAS proteins. We also included some growth factors that are more pertinent to the scleroderma pathogenesis, including TGF-beta. We also have some DNA damage genes put in there since we saw that in our tissue culture phenotype. And so from there, we can generate the score and overlay this gene list onto our single-cell data. The higher the senescence score, the more genes that are expressed in that cluster of cells. And with the statistics you can see in red, the endothelial cells have the highest senescence score and with a very significant p-value. In addition to that, we can also look at our spatial seq data generated by 10x Visium. We know the resolution is not very good for this platform, but you can kind of see these hotspots where there's higher senescence scores in the deep dermis. And also this kind of overlays with our endothelial cells that are annotated in this particular skin biopsy, further supporting that there is endothelial senescence in patient skin. And so once we have that, we went into our endothelial clusters just to make sure that we can see that in all of the endothelial cells that we identified in our patient's skin. So shown here is the clusters that we found for endothelial cells. We identified 7 different clusters. This is the genes that each clusters are used to cluster all the different endo clusters. And then you can see this cluster 6 is lymphatic endo, so it's on its own. From cluster 0, 1, and 2, these are arterial to capillary endos, and these are vanual endos on the top. And I will have the annotation in the following slide. And so overlaying the 68 senescent genes and senescence score onto our endo clusters, you can clearly see, comparing the orange and the green, in each cluster there is significant increase in senescence scores in all of the clusters identified in our scleroderma patient skin, and the statistics are shown here. Since we did this analysis, there was this gene list that got published by the Mayo Clinic group, and they're experts in the field, and they called this gene list SAN-MAIO score, Skin SAN-MAIO gene list. And so this is 125 genes that they handpicked and also validated based on their single-cell RNA-seq from different disease models. And it has a lot of SAS factors and some proteins in there. And so this is a gene list that's validated in multiple systems in human aging models, animal models, transgenic models. And so we thought this is a very nice gene list that is validated much better than our homemade gene list that we did. So what we did here is we reanalyzed our data using the SAN-MAIO gene list, and very happy to say that it is very similar to what we saw in our homemade gene list. And here the clusters are annotated. You can see this is a normal physiological structure of the capillary bed. You start from arterioles, and then you have this capillary loop, and then this is the vanuoles. And so these are clusters in the same way. These are arterial endos, followed by the capillary endos and the vanuole endos, and then the lymphatic endos. You can see besides the vanuole V1 cluster, everything is significantly increased with the senescence score in the patients compared to control. And so we're a little bit nervous about our finding because we know that senescence increases with age. And we did this analysis where we kind of break down the different age groups in our patients and controls, looking at ages at different, breaking down at different ages in 10 years in between. And we compared the senescence score between our patient and healthy controls. You can see that of all of the age groups, the senescence score is still significantly higher in our patient group, suggesting that the endothelial senescence that we're seeing in our data is age independent. Coming back to endo-MT, which I think is a super important phenotype very specific for scleroderma, we're happy to see that we can see this in our single-cell data, where we see an increase in extracellular matrix score in cluster 2, which is the capillary endos. And then this is supported by decrease of endothelial markers in this cluster, also increase in fibrotic markers in cluster 2, similar to what we saw in our isolated cells that I showed you earlier. So this is a very nice validation that we do see this in patient skin. So having the extracellular matrix score and the senescence score together, we can do this correlation analysis where we plot extracellular matrix score on the y-axis and the x-axis is cellular senescence score. We see this mild but positive and significant correlation between the two, suggesting that there might be some link between endothelial senescence and endo-MT, and that's a question mark because we have to kind of work on that a little bit more to be more conclusive. So to summarize and conclude, we showed you that we have the ability to work with these isolated scleroderma endothelial cells from patient skin, and they do retain their abnormal phenotype in culture. Through ATAC-seq, we showed genome-wide changes in chromatin accessibility and transcription factor footprints, and this led us to look at cellular senescence as a whole. We did spend a lot of time validating our findings, showing that cellular senescence indeed presents in scleroderma endothelial cells in patient skin blood vessels. And I didn't have time to touch on the epigenetic aspect of this project, but we do have some data suggesting that SIRT1, which is very important in vascular function, does play a role in mediating, in part, the endothelial senescence we see in this disease. So this is our working model so far. We identified different clusters of endos, and we think that oxidative damage and DNA, oxidative damage, DNA damage, drives this disease of cellular senescence, and this leads to expansion of these senescent endos in our tissue biopsies and in our patients. And through direct effect or indirect effect through SAS, this can contribute to the vasculopathy and fibrosis we see in these patients, leading to the clinical manifestations that patients are suffering from. And perhaps there's room for these xenotherapeutics that we can use to kind of eliminate these senescent cells in patients, and maybe that would relieve our patients from some of the clinical manifestations they're suffering from. And so we recently got a grant to allow us to really dive into the mechanism and also testing out some senolytics and xenomorphics in our system. So we're very excited to move this forward. With that, I would like to thank everyone who contributed to this work, our collaborators from Michigan and outside, and also the funding sources. Thank you.
A great talk.
Thank you.
I'll ask my first question. I'm Slava. For your single cell data, I saw that you pooled a number of patients into your two categories. Do you have enough cells to break it apart and look at patient level data, or could you split the patients in half, those that did better, those that did worse?
Yeah, so this data set is generated in patients that we kind of control it to have, there'll be only early diffuse patients. So it's a very specific a group of patients. So we try to break it down with, more severe disease with the skin score and all that, but because they're all early disease, they have active disease, it's very hard for us to tease that out. And that's a very good suggestion that we should biopsy and sequence more patients to see that, whether we can see that patient and interpatient variability there. Yeah.
Yeah, thanks. Lovely work.
Thank you.
Could you say a little bit more about your senolytic plan? Do you have a specific one or two that you want to study, and how do you plan to take steps forward?
Yeah, so there are a lot of senolytics in clinical trials right now for a lot of disease, right? Aging, cancer. And so the compounds that we're working is actually with Xiaomeng Wang, who is experts in PROTAC compounds. So these are newer degrader compounds that we obtained from his lab and we're going to work with. So some of the targets that we have is the standard BCL-XL targets. Like those are the more common senolytics, but using a more degrader, a more advanced degrading inhibitor to target that. Another target that we'll look at is going to look more at xenomorphics. So xenomorphics are targeting more at SAS. So it's removing these pro-inflammatory mediators that are spit out from the cells. So not clearing out the senescent cells per se, but blocking the effect. And so we have some compounds from his lab specifically for targeting STAT, pathway, for example. And so those are the two targets that we plan to use in our system, using more of an advanced delivery targeting approach than the conventional senolytics that has been tested in the clinic.
I have some question. What is the role of this senescent EC cells in scleroderma? Scleroderma grow in major fibrosis. How is the senescence EC cells contribute to fibrosis?
So you mean, can you repeat CC?
You find the EC senescent cells.
Endothelial senescent cells. Oh, okay. And so this goes back to our model. We think that these senescent endothelial cells is contributing to directly undergoing endothelial to mesenchymal transition, so they're contributing to that fibroblast pool. And also through SAS proteins, there are a lot of profibrotic mediators and pro-inflammatory mediators. We think that we have some data suggesting that these SAS proteins can induce myofibroblast transformation. So I think that's where we think these senescent endothelial cells are contributing, also feeding into that fibrosis phenotype that we see later in the later stage of this disease. Does that make sense?
Do you find any data positive senescent cells in fibroblasts?
Yes, and so if you look at our single cell data, the three cell types that have the highest senescence score is the endothelial cells, the fibroblasts, and the pericytes. So we do think that senescence plays a role in fibrosis too, and I think the pericyte data is super interesting, but we didn't have a chance to look at it yet. And actually, Dr. Varga in our division is actually looking at senescence in the fibroblasts, and we do think that senescence in other cell types is driving this phenotype too. Definitely, it's not just the endothelial cells.
Senescent fibroblasts don't make cars. They are not make cars. How they can make more cars with fibrosis?
In our system, I think we do see that these senescent fibroblasts, at least in the scleroderma fibroblasts, they do produce a lot of collagen. I think it is, at least in our system, I think it's true that these senescent fibroblasts are indeed more myofibroblast-like, and they contribute to the disease. We can discuss more after this, but there is definitely room. We're not saying that senescence is just in the vascular cells. It's one of the cell types in this disease that's contributing to the phenotype that we see, but other cell types definitely play a role here. Yes.
It's a great talk. I just have a quick question. So do we know that whether the cell is entering into the senescence is because of the DNA damage associated with the telomerase? The next question is, can we enhance or overexpress the telomerase expression so that it can maintain and it can prevent the cells into going into senescence?
Yeah, so in scleroderma, we do see that the telomerase is shorter. Telomeres are shorter in this disease. We haven't looked at the endothelial cells in terms of telomeres length. We did think about making our cells immortalized using hTERT and see whether we can expand the lifespan of these cells, but we haven't had a chance to do that.
Sorry, you mean the length of the telomere?
Yeah,
Telomerase is a protein.
Telomere is shorter.
Okay, so telomere is shorter, right?
Yes.
Perfect. Yeah. Thank you.
Mm-hmm.